So welcome everybody. If people want to put in the chat where they are joining us from, and you might also let us know if right now in your region you have active mosquitoes. It's always nice to have a webinar that's focused on mosquitoes because you can ask that question. All right, and we will get going here. So welcome everybody to the 9th Globe Mission Mosquito webinar. And today we're going to be focusing on using NASA observations for mosquito-borne disease. Next slide. And here's going to be our order of go. We're going to start off with a very quick welcome. So welcome. Feel yourself welcomed. We do love having all of you here from so many different states and countries joining us today. We're going to do a very quick overview of the Globe Observer Mosquito Habitat Mapper tool. We're going to talk about how you collect your observations, and then Dr. Ben Zaitchik is going to be giving us a presentation. After that, I will talk a little bit and show you resources for accessing and layering NASA data sets that are relevant to predicting when and where outbreaks of mosquito-borne diseases may occur. Then we'll take a look at the latest Mosquito Habitat Mapper honor roll and then talk about upcoming events. Next slide, please. So, quick review of the Mosquito Habitat Mapper campaign. We're basically bringing together people of all ages to help us better understand the frequency, range, and distribution of mosquitoes. And in particular, we're looking for the 3 genus of mosquitoes that transmit diseases. And one more click. You're helping us to conduct research to see how these vary in response to changes in environmental conditions. And this is a fusion of the formal Globe and the Globe Observer programs. Next slide. There are 4 steps to the Mosquito Habitat Mapper tool. Remember, you do not have to do them all. The first step is to identify where there could potentially be mosquito eggs or mosquitoes that have laid their eggs and be larvae. Even if it is not active mosquito season, it is a great habit to get into, just going around on a regular basis and identifying potential places where there could be mosquitoes. For those of you, and I know I'm one of those people, who have mosquito traps, you don't just look at your mosquito trap, also look all over the place. For me so far, I have not had any mosquito larvae showing up in my mosquito traps. However, I found them in lots of other places. So don't just rely on your mosquito trap. If you do find some, then you do a sample and a count. And then if you're super lucky, and if you happen to have a little magnifier for your smartphone or your iPad, you put it on, and then you can help to photograph and potentially identify the species of larvae that you have. And then finally, super important when you can, to eliminate the breeding site. Next slide. Few things just to remind you, be sure you're collecting data even when it's not active mosquito season. Those are a couple of places where I found standing water when I looked around my house and yard earlier in the spring before mosquitoes started to rear their ugly heads. And you might see this first picture over here where I have a little girl statue. I have to check that on a weekly basis now that it's summer and just make sure that there's no mosquito eggs in there. To the far right, that's one of my mosquito traps that I have up on my porch. And I also check that regularly to see if I have any mosquito larvae. Next slide. Remember to submit your data. It's wonderful for you to take data. Sometimes we're collecting data when we don't have internet, or we want to wait and be ready to submit our data after we've done potential edits. So, just remember when you get a chance to go on and submit your data so that we have that. Next slide, please. We are rocking with the collecting and submitting of mosquito habitat data. As of July 8th, we have a total of 13,951 observations. When we first started this campaign, I was doing a little research earlier today. When we first started the beginning of October, we had 4,730 observations. So between that time and now, we have been making just the most incredible strides with making observations. So thank you guys. Really, we're getting enough data to actually start to have, data that is incredibly useful in many ways to many different people. And if you look at the map that I have there, that was as of 7/8, showing the different locations where this mosquito data has been collected from. Next slide, please. We have a lot of, we have over 19 satellites that are collecting Earth observations, and some of those satellites are collecting data for environmental parameters, which are very important to help us know if mosquitoes may be active, may be in the process of going through their life cycle. And so being able to access some of that data, next slide please, such as precipitation, relative humidity, surface temperature, soil moisture, vegetation, all of those are important parameters to better understand whether or not mosquitoes might be active, and then to also help us understand where those mosquitoes that are active could have more dense populations of mosquitoes. And when Dr. Zaitchik describes his research, and later I'll put a link up where you can access a video that we made of some of the research that he's been working on, you'll see how important it is to be able to collect this information, because even though people may show up at a hospital or a doctor's office and have the symptoms and be found to have malaria, that doesn't mean that's where they were bitten by the mosquitoes. And of course, it's important to know where the mosquitoes are biting so that you can take some preventative measures so that you have either fewer mosquitoes or that you have people that are prepared for and protected against mosquitoes biting them. Next slide, please. So it is my great pleasure, and without further ado, to present Dr. Ben Zaitchik. He is an associate professor at Johns Hopkins University in Baltimore, Maryland. His research includes understanding, managing, and coping with climatic and hydrologic variability. He uses a combination of observations, both in situ and remotely sensed and numerical modeling techniques to conduct his research. So it really is our great pleasure to introduce Dr. Ben Zaitchik.
Great. Thank you, Dorian. Can you hear me?
We hear you. You sound great.
Okay, great. Let me share my screen here. There we go. How's that?
That looks great. Looks perfect.
Great. All right.
All right.
All right. Well, thank you all. And thanks, Dorian, for the invitation to speak to you today. I'm going to talk a little bit about some work that I've been involved with that looks at malaria in the Western Amazon. And so Western Amazon, we're going to be talking about a region that includes parts of Peru, Ecuador, Venezuela, Brazil. About 90% of the malaria in the Western Hemisphere happens in the Amazon, and it's really concentrated in this Western Amazonian zone. And so that's why we're looking there. And as you can see from this opening picture, this is often a disturbed habitat. And so we're dealing with some very dense forests, but we're also dealing with significant sometimes clear deforestation for agriculture and ranching, very often selective logging, but lots of interface between people and the forested or recently deforested environment that leads to high malaria risk. And before I go any further, I do want to acknowledge the fact that I'm a member of a fairly big team working on this. And so this is work that has been supported by NASA and is been led off and on by myself at Johns Hopkins and by Professor William Pan at Duke University. And we also have great colleagues from the University of Albany, from the Health Ministry of Peru, and I should also say more recently the Health Ministry of Ecuador, as well as academic colleagues from Ecuador and Peru. And so here's that graphic that Dorian just flashed through, showing where we are here in the Western Amazon. I'm zooming in here to the Loreto district of Peru, which is an Amazonian district, and it's the place where the majority of malaria in Peru happens. And these bars, again, which Dorian just showed, are our weekly malaria case counts. And the locations of these show the health posts we're getting data from. There are about 300 health posts in all that are reporting in Loreto. And you can see that there's a significant variability in space and in time in terms of the number of malaria cases reported. As Dorian alluded to, just because you get a spike in cases doesn't necessarily tell you where that's come from. And so part of our endeavor here is to figure out if we can begin to use these data in combination with NASA Earth observations to begin to explain and eventually predict those outbreaks. And so a key challenge going on right now in this part of the Amazon is that malaria has reemerged. So here's showing a time series of annual malaria case counts for Laredo, that region in Peru that I was just showing, for starting in 2000. What this doesn't show is that in the 1990s, actually cases were quite high, higher than they were here. And that corresponded to the initial kind of frontier period as people went out into the Amazon and you had a naive population encountering malaria for the first time. So once that first kind of frontier epidemic died down, it had kind of settled out to these endemic levels with episodic outbreaks. And that was happening in the early 2000s. And then there was a big effort by the World Health Organization through the Rollback Malaria Program to try to eradicate malaria from the Western Hemisphere. Now, malaria is not a disease that many people think we can eliminate through standard interventions globally, but it was thought that standard interventions, by which I mean bed nets, behavior control, spraying, not getting to like transgenic mosquitoes or any of the new techniques, that you could get rid of malaria in the Western Hemisphere. The goal was eradication, and they really seemed to be on the way there by the late 2000s. And then a combination of loss of funding for that program and some hydrologic variability, including a massive flood season, seemed to set things back up. And these are final data through 2015 that I'm showing. But if you look at some of the more preliminary data, and there's now detailed weekly case counts showing both Vivax and Falciparum varieties of malaria for Laredo, you can see that since 2012, it's really gone up and stayed up. And so the case counts are now at or equal to where they were in the early 2000s. So it's been really a grand challenge for the region. And this is just Peru. Our colleagues in Ecuador have found that in Ecuador, the case counts in the Amazon region had been very, very low, to the point that they really weren't a concern, and they've gone up as well. And so right now, there's a big effort in the region to figure out what's going on and to try to get ahead of it. And so our question is, can we help? So can NASA Earth observations help both predict malaria and then eventually inform prevention activities? So that kind of question depends on, if we want to answer in the positive, we need to address 3 points at least. First is the claim that the vector species, that the mosquitoes, are sensitive to the kinds of things that we can observe. So that land cover, climate, hydrology. The second claim, which is related to but different from the first claim, is that those variables that we observe, land cover, climate, and hydrology, can contribute to risk prediction. So just because a vector is sensitive doesn't mean that you can actually contribute to malaria risk prediction, right? Related but somewhat distinct issues in that maybe the vector is sensitive, but in ways that's not predictable, or in ways that do not end up influencing the transmission of malaria because of the ways in which humans are interacting with the environment. That leads to the third claim, which is that malaria transmission is going to be sensitive to interactions between individual behavior and the environment. So by individual behavior, I'm really focusing on human behavior, but actually mosquitoes are individuals too. And so their behavior is going to matter. And so what we've been working on over the last several years is trying to address these 3 points in order to develop an warning system. So I should show a picture of mosquitoes since this is the Globe webinar. And so here's our mosquito. This is an Anopheles is the genus that we're concerned with. Okay, so the Anopheles mosquitoes are the primary vectors of malaria in the Amazon. And there are a number of Anopheles species that are competent vectors. Not all Anopheles species are competent. And so you need to get down to the species level to understand if the mosquitoes are going to be a risk factor or not. But we're really focused on that genus. I'll start off with some mosquito data. And just to emphasize the importance of getting these species-specific, or at least genus-specific mosquito observations, these are data that I'm showing here, and now I'm not sure how clear it is on your screen, but I'm showing here a map that has a couple of Landsat satellite footprints in this portion of Laredo that surrounds the city of Iquitos. So the city of Iquitos is right here, and what you're seeing here are some collections that were done along roads going in and out of Iquitos. Iquitos famously is the largest city in the world that you can't reach by road. You have to get there by river or by flight, but there are a number of roads out of Iquitos to some neighboring towns. And so that's what we were focused on, because there's a lot of forestry activities along those roads, as well as up some of these tributary rivers where loggers are active. So we had great mosquito collections there that got everything down to the species level, looked at larvae, looked at adults. And here's zooming into one example. This was just along this road that connected Iquitos to a neighboring town. And then we did a land cover classification on the Landsat imagery. And what we found was that for every 1% increase in cleared land area, there was a 3.7% increase in the odds of finding Anopheles darlingi. Now, Anopheles darlingi is a species of Anopheline that's the most prevalent vector in the Amazon. And so in other words, if we looked at all these collection sites, we saw that in these little ephemeral ponds and puddles, we were more likely to find Anopheles darlingi if there had been deforestation within a 100-meter radius. And now one thing that's kind of interesting about that is that it's species specific. So some of these mosquitoes like it when the forest is cleared and some don't. And so here we have a list of Anopheline species. Okay, so we had species specific mosquito collection. And what I'm showing here is a sensitivity to forest area for each of these species. Okay, I apologize for the acronyms here. There's a lot of these different species here. DAR is darlingi. That's the one that we're most concerned with. But it turns out that all 5 of these are competent vectors. Okay, the ones over on the right side here are not competent vectors. So they're Anopheline mosquitoes that are not going to contribute to malaria risk. And interestingly, as forest area decreases, so as you see a decrease in forest area, you would see an increase in the prevalence of these competent vectors, and you would see a decrease in these mosquitoes that are not a malaria risk. So you clear forest, you get more of the mosquitoes that you don't want from a malaria perspective, which is interesting. So if you just had a total mosquito count, you might find that there were fewer mosquitoes total. But a large percentage of those mosquitoes that showed up are of the species that are going to be transmitting malaria. So deforestation ends up being a positive risk, a positive factor in terms of increased malaria risk. And that's both because the mosquitoes that you don't want are there and because when the area gets cleared, the people are more likely to be there. Okay, so we have that information on land cover that we now know we need to account for when thinking about malaria risk. Second thing we want to get at is time varying risk as a function of weather and water. And so looking at kind of what we just call hydrometeorology, again, the weather conditions and the water conditions, we went back to our NASA toolbox and decided to implement a land data assimilation system. And that's just a NASA term for a tool that is taking hydrologic models, so running a computational model to understand where water might be at any given time, and then combining that with observations. And so as we run the model forward in time, we're taking in satellite data, both to tell us what's happening with vegetation and soils, but also to kind of update our estimates of water storage of vegetation conditions. And the idea is that by combining the satellite data with this kind of process-based hydrologic model, you can get the best possible estimates of what's happening. So it's just our attempt to basically get the best estimate we possibly can of what's happening out in these fairly remote areas where you simply don't have dense measurements of weather or of water. And so to put that in graphical form, what this is basically doing is we're taking landscape information, like our land cover information from Landsat. We're combining with that meteorological data, very often taken from NASA precipitation measurement mission capabilities. We put that into this numerical model, which is a gridded hydrological model that gives us a simulation of what's going on in the soil, with the vegetation, with the groundwater, and solves for the water balance. And from that, we'll get some kind of an output. And here's an output of, for example, a standardized anomaly in soil moisture across a large area to understand what our current moisture conditions are, which might be relevant to mosquito breeding habitat. Because it's a land data assimilation system, as it runs forward, we can update with additional satellite observations. So here's an example of this is just a gridded soil moisture estimate that could then update the model and make sure that it's not drifting off course, that's staying on track with what the reality is. Okay. So why do we do all this? We do all this so that we can get estimates of what's happening in hydrologic conditions. And here's a reason why that might be important. here I am showing again the same range of species, all anopheline species. And this is showing their sensitivity to precipitation in the week before data collection. And we're seeing here, and so if it's black, I should have said this before, if it's black, it's not statistically significant from 0. If it's red, it is. And when we look at precipitation, it turns out it isn't a very good predictor. And that might be because precipitation is noisy. You get a bunch of local storms that are hard to track and hard to quantify. It also might be because we're in a very wet area, and just, when the precipitation comes in a single event, it just doesn't make a big difference. But if we take that precipitation data and run it through our land data assimilation system, we get estimates of soil moisture from our model. And here we have our 5-day average soil moisture, which is integrating information on precipitation and then taking into account the temperature, taking into account how much solar radiation there is, that might be evaporating water, taking into account what kind of vegetation you have sucking the water out of the soil. And so you get an estimate of what kind of water there is, what the water conditions are on the ground. And there we start seeing some significant relationships with our vector species. Okay. And so 2 points here. First of all, going from the satellite precipitation observation to soil moisture by integrating with this land data assimilation system gives us predictive power. Second thing, which is a little surprising perhaps at first, is that these relationships are negative, which means as you get an increase in soil moisture, you get a decrease in these mosquitoes, okay? And I think a lot of us, certainly myself, living in Maryland, I'm a little surprised by that, right? To me, it's like when it gets wet, that's when I expect to see more mosquitoes. But we're in the Amazon here. And what we're finding is that really wet conditions end up flooding out the breeding habitats. And so here you actually see a situation where slightly drier conditions are favored by the mosquitoes because there's still enough water for them, and that way they actually have a chance to reach maturity. Okay, and so, through analyses like those, we feel pretty comfortable that we understand some of the sensitivity of our key vector species to land cover, climate, and hydrologic conditions. So the next question, can that inform malaria risk prediction? And that's tricky to do, right? So the way that we're approaching this right now is that we've realized that you have to view Laredo District, which again is the area that I'm showing here, as being a collection of ecoregions. So this is all kind of, generally speaking, within the Amazon forest, you have different types of forest. Some parts of the Western Amazon have standing floodwater through much of the rainy season, others do not. Some have a different vegetation type that you find just slightly up at higher elevations. You can take this information, as well as information about the seasonality and the amount of the rainfall, and define these districts. So in this case, we have 7 ecoregions. Then we combine that ecoregion classification with the data we're getting from our health posts. And then we have to estimate human population. And that's a big, big deal that people like me often gloss over, right? You need to know how many people are there to know whether you've got a high percentage of cases or a low percentage of cases. And knowing what the percent population served by a health post in the Amazon really is, turns out to be really hard. And you need that for your denominator, right? Because you need to divide by that to understand what the rate of infection is. And so we've been doing a lot of work on that. Using Earth observation, nighttime lights data can be very helpful. Data on selective logging activities and road networks from high resolution data, trying to find human activity. Then we try to fit a regression model that takes advantage of the hydrometeorological information from our land data assimilation system and our land cover information, and tries to predict these infection rates over time. We do that retrospectively to fit the data, and then we go forward and we try to do 12-week forecasts. Those forecasts aren't weather forecasts. They're based on the lag time between how the weather behaves and how the malaria gets transmitted. And so we can go out to about 12 weeks because current weather conditions have some memory into what the soil moisture state will be for at least a week or 2 in the future. It takes some time for the mosquitoes to mature. It takes time for them to go out and bite people. It takes time for those people to show up at the clinic. And so that's how we're getting our forecast. And so here's just an example of some results for two of the ecoregions, ecoregion 1 and ecoregion 6. Ecoregion 1 is up here. It's kind of some fairly remote forest area along rivers going into neighboring countries. Ecoregion 6 is near the city of Iquitos, and so that's where the densest population is. And what we see is that there's a seasonality that we can kind of capture with our model. Again, the dots here are the actual reported data, and the lines are predicted. Both the seasonality and even more so the year-to-year variability are different by ecoregion. Okay, so you can see that you really do need to look at this separately. You can't just issue a forecast for malaria in the whole region. And then as you look forward to, like, for an example of a forecast here, and this obviously is a forecast from quite a while ago, but I'm showing it because this is when the cases just began to spike again. So that's when we really started looking at doing these forecasts. You'll see there's an uncertainty range, and that has to do with the uncertainty in the model when you're forecasting forward. Okay, and so what we found is, and that's still work in progress. We're working with the Health Ministry of Peru to see how we can operationalize that. But already we're seeing that, yes, we can get predictions. First of all, that show some skill. So we're satisfied. Well, I should say we have significant predictive skill. We aren't satisfied. We're still trying to improve it, but it's better than what they have right now. And another point that I was not aware of before starting this work was that the forecast is helpful, but even the monitoring is really helpful, because at present, the only way that the ministry knows when there's a malaria outbreak is when the cases start to increase. And it takes 4 to 6 weeks for those cases, case counts, to make it in from the health posts and be integrated, quality controlled and put in their system. So by the time they see that spike, it's already gone on for a month or two. With this system, as long as we can kind of project forward, project into real time, we can give them a head start. And that's been they're very interested in that capability, even before you talk about forecasting into the future. Okay, so the last point I want to make here is that up till this point, I've been talking about how you use Earth observations to inform estimates of land cover and weather and hydrology in order to predict where mosquitoes or malaria might be. But implicit in that second step, in figuring out where the malaria is this idea that there's these interactions happening between people and their environment. And up until now, what I've showed you has not accounted for that in any explicit way. It's implicitly there, right? If we have a skillful model, then it's somewhere there, it's accounting for the fact that as the weather changed, people were being put in a position to interact with mosquitoes. But we really want to understand that a little better in order to talk about how different interventions might play out. And so to do that, we implement an agent-based model. Okay, so an agent-based model basically consists of agents, right, which can be people, and as I'll show you in a second, they can also be mosquitoes, they can be plasmodia, interacting with an environment given certain decision rules. And so it's a bottom-up approach, as opposed to kind of the statistical top-down approach that says, hey, we have elevated malaria, let's fit a statistical model to figure it out. We say, no, let's talk about how behavior might lead to malaria prevalence. So for example, here you have a household, and perhaps one member of the household is infected by malaria. And you have other households in the village, some also with people who might be infected, and they might be symptomatic or asymptomatic. We would treat those two people differently. You've also got hydrology, like maybe there's a river running through the village and there are puddles forming, so there are breeding sites. And you've got temperature conditions that are varying, so that's all kind of your physical environment. We have our land data assimilation system that's allowing us to look at things like soil moisture and river level over time. So that's giving us a sense of what's going on with the climate here. We also have temperature from the LDAS. Then we've got mosquitoes flying around, and some of these mosquitoes are carrying, and some of them are not. And then those that are carrying, they're plasmodia, and those plasmodium are at different points in their life cycle. And so all of these, the people, the mosquito, the plasmodia, they're all vectors. They're all agents in the agent-based model. Also, you might have several villages and people traveling between villages and spreading malaria that way. So you have to account for all these levels of interaction. So in our ABM, here's who we got. We've got our mosquitoes, our humans, and our plasmodia, and we've got our environment. The mosquitoes are characterized by certain habits, by how long they live, by how they move. People, some of them have immunity. They also have their own behavior and their movement patterns. And the plasmodium has its incubation times and its transmission efficiency and perhaps resistance for recurrence. The environment is everything we're getting on the natural environment plus the human environment. And so here's the first implementation. What you're seeing here is an example from one village in which we implemented the agent-based model. It's along a river. Down here along the riverbank is where you have the ovipositions happening, because here you've got, again, the kind of breeding sites on the edge of the river. You also have some breeding sites that are located away from the river in different little ponds and puddles. And we are then predicting the number of ovipositions based on the agent behavior of the mosquitoes. We then have malaria cases being predicted, and these are happening in households. And so each of these dots is on a household, and that's based on humans interacting with the infected mosquitoes. And as the environment evolves, that will change the frequency. So why do we do this again? So we can do this, we can predict forward how many malaria cases happen. Evaluating that is really, really difficult. Okay, so the statistical model is very empirical. It's very much grounded in the data, and we can run objective metrics to say, we've done well or we've done poorly. An agent-based model that's trying to figure out which individuals are infected is really tricky. Yes, we can see if there are hotspots in a village, but actually getting down to say, we aren't actually literally trying to predict, that one particular individual is going to get malaria. We are using these individuals as exemplars, as agents that are behaving in the way those people behave. Okay? So we aren't really doing this to predict. We aren't doing this so that we can go to a house and say, watch out, one member of your household is predicted to get malaria tomorrow. We're doing this because it allows us to say, well, what would happen if we tried certain interventions? And so here's an example of that. Oh, and my animation isn't working quite properly. That's okay. And so what I'm showing here, sorry for the little graphical error here, but just if you could focus on this annual average malaria incidence happening up here. And so we've got falciparum, vivax, and total. And what we're doing here on the x-axis is saying, well, what if we do larval habitat control? So in other words, spraying for mosquitoes. And if we spray for a certain radius around the occupied part of the village, okay? What do we predict will happen for the average annual malaria incidence? And then you can figure out, or you can at least predict how much of a spraying area you need, 50, 100, 150 meters or more around the dwellings, if you want to knock down total malaria cases to a certain level. Okay? And so the way that the ABM is dealing with this is basically saying, well, if you're doing larval habitat control with a certain % efficacy, which breeding sites are you knocking out? How is that going to influence the number of mosquito agents you have, the number of infected mosquito agents you have, and the number of human interactions with infected mosquitoes, and therefore the transmission risk? And so you can't really do that with a statistical model, but with this kind of bottom-up ABM approach, you can start to look at that. And here's the graphic that should have appeared later. So then we take this and we say that was for one village and looking at control strategies. Then we say, well, what if we go here and look at the Napo River, which is along which there are a number of settlements and logging camps, flows down and meets the Amazon here just downstream of Iquitos. Napo River also flows in from Ecuador, so it's a transboundary river, and there's a lot of malaria around this corridor and transboundary transmission. So along the Napo, we have our farms, we have our households, and so people are moving between their household and their farm fields. We also have logging camps that people are going to seasonally. And we have various social places where they might be gathering at certain times. So our human agents are moving between these activity centers. Our mosquito agents, meanwhile, are laying their eggs, they're having their life cycle, some of them are carrying malaria. And something we found here is that if you look at all these villages along the Napo River, you can say, well, what's the malaria incidence going to be as a function of the % of workers that travel between villages? And so that's critical, right? Because when it comes to malaria control, you need to understand not only how you're controlling your localized risk, but also what the risk is of malaria coming in from somewhere else that you haven't controlled, or whether you have to expand your control zone. And so here's a case. So in these blue squares here, what we're looking at is % of traveling workers if there's no one that's asymptomatic. So if you know every single person who has malaria because they're showing symptoms, what would your incidence rate be if you attempted certain control strategies? And what you find is that the more % traveling workers you have, the higher the incidence, all right? So as people travel and interact with more environments, perhaps not surprisingly, you end up with higher rates. And this line here simply shows that, okay, if we think about up to 15% of people traveling, which is about what is understood to be the case, you would end up with this kind of malaria incidence. This is an even harder problem, though, because people are not always symptomatic. So if you account for the fact that a lot of people carry malaria and are asymptomatic, you find that as percent of traveling workers increases, you still end up with this increase. It's a more gradual increase, but it's a much higher incidence level in general. And so we find that, okay, we would end up with this malaria incidence that's about double or more than what we have in the asymptomatic case. And it turns out that that pretty much matches what's observed. So the observed incidence rate is right here in the black line. And so we can check again, from the top down, we can check and see if the agent-based model is showing us something realistic. Seems that it is. We're hitting the average incidence rate across the study area. But what the agent-based model allows us to do that we could not get simply from observing that incidence rate is an understanding of how something like traveling between towns and how different control strategies that take that into account might influence future infection rates. So that was kind of the last thing that I wanted to cover today was this idea that you need to take into account these interactions between individual behavior and the environment. And so in that instance, the Earth observation is still very important, right? Because it's giving us information on the physical environment. It's also helping us to understand population movements in some cases. But you have to integrate that information. You have to bring it together with this information that you garner in communication and in collaboration with your communities through household surveys, through interviews, through discussing control strategies in order to figure out exactly what the local dynamics might be and therefore what intervention strategies might be useful. So that's all I will say. So thank you very much for your attention, and I'll turn it back to Dorian to see if there's time for questions.
Thank you very much, Ben. That was just really interesting. It's always very interesting to see how this research is done, and then also how the research is applied in a real world setting, such as helping them to know where to where they're actually becoming infected by these mosquitoes. So do we have any questions? I think we may have had one from Claire, or she might be typing her question in. We'll give just a minute here and see if we have questions, and if not, we'll move on. There should be audio. Oh, Claire, if you type your question in, there you go. Now I think you have audio as long. There you go, Claire. You're good.
Hi, Dorian. Can you hear me?
We hear you. Thank you.
Great. Ben, that was really interesting. Thank you. I have a question about scalability. So much of your data is remotely accessible, and I'm curious about what the data that is not, specifically the information about villages, how that could be kind of estimated from a distance in order to be able to scale up this type of model.
And I'll let Ben answer that because he's had experience actually working with those types of data.
Yeah, and so some of the scaling ability question of the ABM is a big one. And that's kind of what we're working on right now in taking it from one village to several villages. We're just beginning to try to do that. We found that, in so much as you're dealing with similar types of communities, it scales. As soon as you move into new kinds of behavior, new kinds of environments, it doesn't. And so an ABM is, as your question implies, harder to scale reliably than something that's just a physically based model because it depends on some of these very culturally and geographically specific human behaviors. But the structure can scale. And so the ABM that's used here can be applied somewhere else, but you're going to have to go and really ask the hard questions of what your assumptions are on the individual's decision rules. Does that address the question?
It does. Thank you.
Hey, Ben, I see there's another question. It says, do you believe we can have maps with mosquito disease prediction information made from AI using globe data for some countries around the world? And you may not be able to answer that since he is, I guess, talking about globe data.
Yeah, I mean, yeah, you're right. I don't know the details of the globe data. when it comes to AI and these problems, I mean, in principle, yes, right? I mean, if you have a large enough data set and you have enough information, and that data set is representative, there's no reason you couldn't use some kind of machine learning algorithm. Our statistical model is simply, so much machine learning, right, is simply another way to do statistical modeling. And so if you want to say by artificial intelligence, we're talking about doing something like, mapping out risk with a convolutional neural network or something like that, sure. Structurally, you can do it. It's all a question of whether the data supports it. Getting from the globe data on mosquitoes to the disease would either require another very rich data set on the disease of interest, so you could then train that algorithm to it, or would require that you combine some kind of machine learning based mosquito estimate with a different type of risk analysis, right? So I think that you'd want to figure out the most appropriate place to apply that algorithm in the value chain.
Thank you very much. All right. I'm going to go ahead and move on so we make sure that we get the next part in. If there are more questions, please feel free to take a look at the chat box. And I know that Dr. Lowe's been busy working away on that. And Ben, thank you again. Really, really appreciate you making some time to work with us today.
Great, thank you.
You're very welcome. Well, what I'm going to do next is I'm going to kind of quickly show an example of how you could take the case study that Dr. Zaitchik was talking about, the malaria in the Loreto region of Peru, and show you how you can use Giovanni to access NASA precipitation data, and then how you can use Worldview to access data that would give you information on the temperature and also on humidity. You also can use Worldview to access soil moisture, vegetation cover, ponded areas, but there's not time within the scope of the next 10 minutes to be able to cover all that. So I'm just going to kind of take you through very quickly how you could go into these tools and work with them. Most likely you're going to want to go into these tools, and I put the links up there for you, and play with them. You cannot break them. Get in there and play with them. They're free tools for you to use. And so let me just go ahead and give you a little bit of introduction. Next slide. So what we're doing when we're talking about these different variables that perhaps are having an impact on the number of and when you're going to be seen. In our case, active mosquitoes. Somebody had a question earlier talking about, well, why aren't we looking at say dragonflies and other types of insects? And in that case, I think he was referring to the fact that dragonflies can eat mosquito larvae. What's really neat about this type of data that I'm going to show you is you can use it for all kinds of purposes. I think that for those of you who are doing some research and potential science experiments, you could be taking a look at using these tools I'm going to show you, what is the satellite precipitation estimates for your region? What is the humidity data? What is the temperature data? And you can also be collecting that in situ. So, using your globe protocols, using your instruments, and then compare and contrast that data and see if you can determine a cause and effect link between what you're finding with your data, such as your temperature, when your temperatures are at certain ranges, and what the NASA observations are showing you, and then compare that with maybe some data that you're getting yourself. So next slide, please. In the case, and so these are what I just put in the chat box, so you can access those. Giovanni is what we're going to be using for the monthly precipitation. Now this is rather small, so you probably can't see it too well, but if you were to go on to the Giovanni site, you're going to see that you have some options along the top here, and here you're kind of deciding what type of data you want to be working with. And then down here, you are making some other decisions about the date, time, and then what your keyword is, the specific type of information, environmental parameter data that you're looking for. Next slide. So in this case, and I would fully anticipate you are not going to absorb everything I'm saying right now. These slides will most certainly be made available to you so that you could go back and you could take a look at this. Cassie does post our final webinar on the website. And also, if you write to me, I'll be happy to send you just the slides so that you can go through them. But basically what you're going to do is under keyword, you'll go ahead and put in iMerge final and do a search. You're going to get some different options showing up there. You're going to pick merge satellite gauge precipitation estimate. That's the kind of data you want to work with. Final run. In this case, we're going to be looking at monthly precipitation data. You can also look at precipitation data daily. You can look at it in other ways. But for the research that you might be doing, if you want to be looking at what the impact of precipitation is on the amount of mosquito larvae you find, doing it on the monthly scale would work perfectly well. For selecting a plot, you go into the time series and you select seasonal. Once you open up seasonal, you have an option of which dates you want as far as which months you want. Go ahead and click all the boxes. You want all the months. Setting the years, as Ben was saying, we noticed that there was a huge increase in the malaria cases. Actually, starting in between 2012 and 2013, we started to see that huge uptick. In using this as a case study, I thought, well, you could go ahead and look at, say, 2014 through 2017 and see what you find. To select your region, what's nice in Giovanni when you are looking at your seasonal data, your monthly seasonal data, is that you can just enter your coordinates instead of trying to work in trying to go into the map and find your exact location. In this case, I looked up the Loreto region. I used Iquitos as the sample place. And what you do when you're working with Giovanni is you enter your longitude first and then your latitude, and you enter it using decimal coordinates. So that's important to remember. One time I was doing this and I ended up in the middle of the ocean and wondered why was that happening? Because I had entered my latitude first. So in any case, you enter your longitude and your latitude, and next slide, please. So here's what I end up with. When I enter that information, up here was my time series. I selected seasonal. I selected those different months. These were the years I selected. I put in the longitude, comma, latitude up here. Of course, you can have copies of the slides, definitely. Just email me. And then when I did that, it came up giving me this little area here. Next slide, please. And so then what you can do is you go ahead and you press plot data, and it usually takes about 3 or 4 minutes. Maybe you can practice guitar while you're waiting or answer some emails. So you wait and it will tell you that your data is being analyzed. The plot ends up being really interesting. So here we're looking at, of course, years along the bottom. We're looking at amounts. In this case, I asked millimeters per month. You could also pick inches per month if you wanted. And each of these is telling us how much precipitation is falling for different months. So each line here will be following a different month. Now, that's useful. If you click on the exact dot, it will give you the amount. But a little trick that I found, next slide, please, is you can ask it to go ahead and give you the download. So you're going to click on the downloads over on your far left. You're going to have a lot of choices. The choice you want to pick is the one at the very bottom that says your combined ASCII. And now what's going to happen is it's going to give you the specific numbers for the years and for the months in an Excel spreadsheet, which then, of course, you can manipulate to use a little differently than what we have here. I've just finished making a resource that will take people through how to access this data through Globe and then how to access it through Giovanni. So if they want to compare and contrast what you're finding with your Globe instruments to what you're finding with the Global Precipitation Measurement Missions data, then you will have that activity. So I should have that ready in the next week or so. And next slide, please. So that is how you use Giovanni. NASA Worldview, I used to think was easier than Giovanni. Now that I've gotten more used to using Giovanni, I actually find that easier. I'm going to say with NASA Worldview, you definitely want to do the tutorial. So take some time, work with the tutorial. I'm also going to put up here, there are a couple of websites, of webinars that were completely devoted to teaching people how to access these websites. in a much slower fashion to help you better predict, monitor, and respond to both vector-borne and water-related disease. So at your leisure, go ahead and watch those webinars, and you can go ahead and have Worldview and Giovanni up and do those, use the information in Giovanni and Worldview while you're watching the webinar. So when we're looking for our temperature, what we want to work with here is our surface skin air temperature. We're using the data from the Aqua satellite mission. We're using the AIRS instrument data. And if you listen to that webinar, the beginning webinar, you'll find out why we're using that data and a little more information about Aqua. So in any case, we're going to be picking the skin surface temperature. Day and monthly is going to be what we're interested in working with. And I'll show you how you do that. With relative humidity, we're also going to use Aqua, the AIRS instrument, and we're looking at surface relative humidity day and monthly. Next slide, please. Yeah, kids will learn how to do this intuitively. There's different ways you can interact with Worldview. I like to start off by setting the date to where I want it to be, because sometimes I'll go to collect the data and I won't see it show up, and that's because it always starts off at the current day and time. So you go down to the bottom there. I went ahead and I set it up to begin in 2014. Also, I went over to the little side up here and I set it so that it would tell me about months rather than days, or so that's what I'm interested in. You can set it for years, months, days. So I set it so it would begin on January 1st of 2014. Next slide, please. Now with this one, you don't have the option of entering your lat and longitude, but I would suggest, because it can get a little tricky when you're in here trying to figure out with the maps, I suggest you go ahead and figure out what the lat and longitude is. In this case, I just looked it up, again, working with your decimal equivalents. And then what you can do is, once you're in the map, zoom close to where you know. So I knew that it was near the borders in northern Peru. So I came over and then I just kind of moved my cursor around, and then it told me when I was at the latitude and longitude. Kind of reminds me of the old battleship game where you're moving it around to try to get your coordinates to line up. You don't have to be exactly on the spot at 3.749. Actually, it's minus 3.749. The minus stayed up there, and minus 73.254, but just somewhere in the general vicinity. So you're kind of honing in there. So we're selecting, we've got our dates, and now we're selecting the region that we're interested in. And next slide. So then you go down here, you've got some red buttons. You're just going to go ahead to, a red button, and you're going to push add layer. Then it's going to give you a search box. In the search box, I said I wanted to search for surface skin temperature. And again, I'm looking for the day. And you can have day temperature and night temperature. In this case, just to keep it consistent, I chose day. Mess around with it, play with it, try out different things. I picked monthly, Aqua, and AIRS. Now the results are going to come up in Kelvin. So I looked up to find out what the results should, if I wanted to take 18 to 32 is the prime temperatures for the Anopheles darlingi, which is the most responsible for malaria in this region. But as Dr. Zaitchik said, we're going to find that there are, different temperature conditions for different species of mosquitoes. So in this case, anything between 18 to 32 would tell me that the temperatures are right for there to be active mosquito breeding habitats. So you can get in here and you can change the palette to a different palette should you so desire. I used for this one, I stayed with the rainbow, the default pattern palette. You can change the opacity, and you can also change the threshold, just depending on what it is you want to do. In this case, I did change the threshold so that I was looking at temperatures. And again, this was in Kelvin, so I set it to where it was about 260, 275, and up, just to get a feel for the colors that were then going to show up were going to be indicative of regions that could potentially have active mosquitoes based on temperature. Next slide, please. And then you can do the same thing with relative humidity, going through that same process. In this case, the threshold generally for Anopheles darlingi is about 60% relative humidity, so I set it for 50. And again, work with a different color palette that meets your needs. Sorry to rush, but next slide, please. So, as I said, there are a couple trainings that you could access that are useful so that you can learn more at your own leisure, not having to rush through like I was doing. I also put on there where you can see some of the different recordings of some of the training resources that are accessible that were from a workshop that we did where we had different people coming in and talking about how you could use NASA Earth observing data for vector-borne and water-related diseases in general. And next slide, please. So there's some other resources. When you have these slides, all of these hyperlinks will be clickable, and I also put those up there in the chat. And next slide, please. Finally, I just want to call out our top data collectors for the past 30 days. And you can see here, we've got a lot of countries, but I'd like to see some more observations coming in from the USA. So these are now not the only people that have sent in observations, but these are our top people for the last month who made the top list every single week for the last month for sending in the most mosquito observations. All right, and next slide, please. Finish up with coming soon to a webinar and a newsletter near you. So next week is the Globe annual meeting. I hope I'll see some of you there. Our citizen science webinar will be on Wednesday, July 24th at 2 p.m. And how can we use mosquito citizen science data to protect from disease? And then Wednesday, August 7th, we're going to be going around the world with Mosquito Habitat Mapper and learning how it's being used with different people from different countries around the world, as well as getting a feel for some of the landscape and the geology around the world. I figured summer's a time for travel for many folks, so we'll have a little armchair travel with that webinar. So for everybody, thank you so much for joining us. We'll stay on for a little bit here and see if there are any more questions. And sorry to be rushing things toward the end there. And again, a huge thanks to Ben for presenting for us.